Hello, everyone. My name is Chenfeng Ke. I'm an associate professor of chemistry at Dartmouth College. I would like to share with you about my research funded by the Arnold and Mabel Beckman Foundation. My first slide is really dedicated to Dr. Arnold Beckman. His inventions, such as pH meter and spectrometer, revolutionized the laboratory instrumentation and change the way that we measure and analyze chemical reactions. And this really opens up new avenues for scientific discovery, including my own research. Particularly, Dr. Beckman's legacy as a scientist, innovator, philanthropist, as well as his relentless pursuit of knowledge, shed light on my own career. So my research group is currently funded by the Arnold Marble Beckman Foundation to conduct fundamental research in developing functional materials for 3D printing applications. In particular, we're trying to address fundamental problems in transfer small molecule functions to the microscopic scale in connecting with the additive manufacturing techniques, also known as 3D printing. So on the right, you can see a cartoon representation of a 3D printer. And in the middle, you will see a video in action to show a 3D printing extruding a hydrogel and building a lattice cube that is funded by this research. At a fundamental level, we aim to develop stimuli-responsive, self-healing, strong and robust, recyclable materials that will enable us to have a sustainable future. The question is how we are able to transfer the molecules and order them to tell them to march in the same direction that allow us to transfer their molecular functions and motions to the microscopic scale. And this is not a trivial answer to address that. So in order to do that, we have to think about ways to organize these molecules in a synchronized manner. In particular, we are also interested in functional 3D printing materials development. In this particular case, we need to also consider when we're introducing molecular entities into 3D printing gels, they need to possess the viscoelastic hydrogel properties for extrusion. And this allows us to introduce a variety of supermolecular binding motifs. For example, hydrogen bonding, host gas interaction, metal ligand coordination, electrostatic interaction. And my group particularly pioneers the microcrystallization methods and van der Waals interaction in enabling functional molecules to transfer to functional 3D printing materials. Funded by the Arnold and Marble Beckman Foundation, my research group developed 3D printing hydrogels, particularly using microcrystallization approaches. In one example that I would like to share with you is we functionize a traffic regulating functional groups on a long polymer axial chain. In this case, we're able to control the molecules threading and translocating on these polymer axials. An analogy would be thinking like you are driving your car in a highway, but we block one of the exits. In this way, you need to take detours, and these detours change the molecule property at a microscopic scale because everyone needs to follow a different traffic route. As such, we are able to change these so-called polypseudotaxing threading and crystallization behaviors and converting a traditional, not very useful precipitate into a functional 3D printing materials. And we're using these 3D printing materials to trying to understand fundamental questions. For example, in the literature, people trying to understand how does this microcrystallization happens. In this particular research, we obtain a single crystal structure that allow us to atomically understand how these molecules arrange in relative to each other. And this information really changed our perspectives and importantly, correct the misunderstood packing models that is existing in the literature for the past 30 years and allow us to unambiguously unveil the fundamental chemical informations of these polycellular taxanes align with each other. And understanding this molecular arrangement is particularly important that allow us to transfer these functions to the microscopic scale. And taking this approach, we're able to fabricate a variety of 3D printing hydrogels that we can print nodes, mimics, lattice, and pyramids. And after using different inks and at different temperatures, we'll be able to convert a same starting ink formulation to a variety of different materials. And using this feature, essentially, we can using one ink or one ink formulation to create different materials using different 3D printing heads and while printed at different temperature. And this is a showcase of the 3D printed design that would print these hydrogels at 60 degree and 20 degree. And this allows us to program their shape changing upon exposing to moisture. We also printed a flower, a 2-layer flower, and exposed this to moisture. Upon moisturizing, you can see this flower closes up. And if you dries out air, and this flower will open up again. We also designed a mechanically robust hydrogels that we can threading cyclodextrins and convert a very soft hydrogels gradually to a relatively plastic and more plastic materials. And if you wash out these cyclodextrins, they become elastic again. Essentially, we can dynamically tune a material from a rubber band to a hard plastic. And using this feature, we 3D printed a sea cucumber mimic, the soft and hard regions of a sea cucumber that after adding cyclodextrin, the papilla mimics area become very stiff that they can protect them against predators' attack, while the soft area allows them to insert them between the gaps of rocks to hide them from being attacked. So this is a very powerful way to generate biomimicry materials by combining molecular design and 3D printing technology. Because the molecules and polymers we are using, cyclodextrin and polyethylene glycol, is biocompatible, we also find this is a great engineered scaffold. By licensing these hydrogels developed in my group to a startup company called Curans, we were able to show that by implanting these hydrogels to the mouse brain, these hydrogels can help the mesenchymal stem cells to differentiate into neuronal cells. And we'll keep working on this direction with the hope that these type of materials will provide different mechanics to allow us to mimic different tissue stiffness. Last, I would like to thank again the Arnold and Marble Beckman Foundation in supporting this research, which has transformed our understanding of these polycellular taxanes and enable us to develop a family of biocompatible hydrogels that are potentially useful for biomedical usage.